small molecule chemical tools for IDG dark kinases with particular interest in neurodegenerative diseases.
Thank you. Thank you for the introduction. I will try to keep on time. So I'm going to shift gears from what Sean has just talked about. The kinases are very well known for oncology, but I'm leading an effort looking at kinases in neurodegeneration. So to start, I just wanted to show you the landscape for kinases in neurodegeneration. They are studied. There's ongoing clinical trials for either between 10 to 30 clinical trials actively going on in, if you go to clinicaltrials.gov. And I've shown some examples here for ALS, Parkinson's, and Alzheimer's disease. And what we see is most commonly they're the more well-studied kinases like mTOR, LRRK2, and p38 alpha. But there are studies going on with kinases in this space. So what I wanted to do was look at some of the dark kinases, the IDG kinases, to see if these are implicated in neurodegeneration. And so I published a review earlier this year, and here I'd like to highlight my findings with respect to ALS. So looking at the IDG kinases, I did some text mining exercises and identified several kinases, 9 in total, that were implicated in ALS, either via a GWAS or that they were overexpressed in patient samples. Then moving to the right, the table there shows either what the kinase variant mutant, what the phenotype is in the patients themselves. You can see down in the bottom left that these 9 kinases are not they're not co-located; they're across the kinome. And further, I went into the GEO database, and from these 9, 7 are pretty well expressed in either neuroprogenitor cells or in fully differentiated motor neurons. So these 7 are the ones that I'm focusing on. So how we're doing this is I'm working with the UNC Pluripotent Stem Cell Center. We take either embryonic stem cells or induced pluripotent stem cells, gene edit out the kinase of interest. The entire coded region is deleted through CRISPR. Then we differentiate them to motor neurons, which takes about 40 days. During the differentiation, we're looking for changes in rate, markers, or attachment of the cells. And then finally, the fully differentiated motor neurons, either with a knockout or wild type, are looked at in terms of if they cause protein aggregation, if there's differences in morphology, response to stimuli, how they act, basically. And what we hope to do is to follow this up with some of our high quality small molecules. So I'm a medicinal chemist. We make kinase inhibitors that target these kinases. Do we see the same phenotype? Do they genotype copy, or do they act differently? And are these therapeutic options for these diseases? So moving to the next slide, this is where we are right now. For 6 of the 7 ALS implicated kinases that I've discussed with you in the table there, we have knockouts confirmed in iPSCs. NEC1 is the one that's remaining to be done, but should be done soon. Moving forward with PXK, one of the kinases, we've actually taken it to the neuroprogenitor stage, and it's well behaved. So the differentiation is ongoing to fully differentiated motor neurons, which we'll observe as that differentiation is happening. We're to use those PXK knockout neurons to standardize our staining protocols. So we're looking at specific readouts, which I'll summarize in my last slide, but basically we want to use these to see how our cells respond to different antibodies that we've identified for immunohistochemistry. And in parallel, we're also treating iPSCs with some putative inhibitors of these kinases in the table that we've identified through the IDG program, and we hope to then follow up with the knockout with these small molecules treating the cells to see if we see the same results. So looking toward future directions in the next 2 years, which is and beyond, the remainder of the IDG portion of the project. So we have chemical tools that are in various stages of development. So for 2 of the kinases in the red box, these kinases are very poorly studied. There are no known commercial panels that have screened for these kinases. We're developing the nanobret assays, so the cell-based target engagement assays, but we really have to start from square one in order to identify chemical matter to target these kinases. In the middle box, we have kinases where we have nanobrets developing. There's chemical precursors we've found by combing the literature, and they're being optimized basically to get to this level of a DK tool, a dark kinase tool, or optimally a probe compound. And then we move to the right and we have the green box there where there's 2 kinases where we're at a very advanced stage. So with these 2 kinases, we've identified a DK tool for DYRK2. It's very potent in the cellular target engagement assay. And for CK1G3, we have a very potent tool against the whole CK1G family. And so we anticipate that it will be active on this kinase. And then moving forward to the box at the, below, you can see kind of what we're hoping to do with the antibody staining like that I mentioned previously. So I'm looking at differential effects on cilia, DNA damage repair, and protein aggregation. And we have specific antibodies that are going to be targeting cilia growth and branching and how the length of them, looking at motility of the cells, seeing how they respond to DNA damage, either through a chemical protogen or irradiation. And then finally, looking at protein aggregation. TDP43 is one of the most commonly aggregated proteins in ALS, and as well as other neurodegenerative diseases. So we want to see how the kinase knockout impacts the aggregation of this protein. And we're going to look at our knockout cells versus a cell line, an IPS cell line where TDP43 is actually knocked out. So I'm a little bit short, but I think for the sake of time, I'm going to stop here and I will take any questions via the chat.